Thank you very much for the opportunity to speak at this exciting symposium today. And the title of my talk today, as shown here, is quite broad. However, given my short presentation time, I would like to focus on using block polymers to transiently coat the inner wall of liver sinusoids. Okay. So, as shown in this slide, there are four main challenges when delivering nucleic acid therapeutics systemically to particularly extrahepatic targets. So today, I will concentrate solely on the first challenge, the preventing the capture by the reticuloendothelial systems during the systemic delivery. To avoid capture by the RES, it may be necessary to design what are known as stealth carriers. So as shown in this slide, the stealth carriers we have developed so far are polymeric micelles formed by the self-assembly of amphiphilic block copolymers in water. The core of the micelle here serves as a container for the drug, while its exterior is covered by hydrophilic and flexible polymer chains, such as polyethylene glycol shell, which enables it to have stealth functions. Various forces can drive core formation, but electrostatic interactions are particularly important for nucleic acid therapeutics. Well, the liver macrophages, or Kupffer cells, located on the blood vessel walls, are believed to play a major role in capturing nanocarriers in the liver. However, the entire liver sinusoidal endothelial cells also play a crucial role in nanocarrier capture. This will be demonstrated in the following video of in vivo confocal microscopy of a living mouse liver. Okay, here, we are now observing the sinusoidal capillaries of the liver under the in vivo confocal microscopy. When we administer non-pegylated DNA polyplex, a non-stealth nanoparticle, through the tail vein here, you can see micron-sized non-uniform aggregates form immediately and are stuck to the capillary walls. Subsequently, when we administer polystyrene particles labeled with a blue fluorescence dye, a marker for Kupffer cells actually, they highly co-localize with these red aggregates, showing that these are captured by Kupffer cells. Now, let's look at the results for polyplex micelles coated with polyethylene glycol. As you can see, no aggregates form this time. The PEG functionality is inevitable. Also, there is hardly any co-localization with the blue Kupffer cell markers. And this figure quantitatively shows the pegylation significantly reduces nanocarrier co-localization with Kupffer cells. This is good. However, as seen on the right side, there is still adsorption to the entire sinusoidal wall, even with pegylation. So this indicates the liver sinusoidal endothelial cell mediated capture is still a big problem. So as illustrated in this schematic, for stealth nanocarriers, the issue is not only capture by Kupffer cells, but also adsorption and uptake to sinusoidal endothelial cells is a problem. To solve these problems, further enhancing the stealthiness by modifying the surface structure of nanocarrier is conceivable. However, it's difficult task actually. So here, we consider the alternative approach. That is stealth coating of the sinusoidal endothelial layer itself. In other words, temporarily covering the sinusoidal wall with a polymer, such as polyethylene glycol. So we designed a block copolymer for a transient coating of liver capillary walls. Well, there are two necessary conditions for this vessel coating agent. First, the coating should be transient, as long-term coating may impair the physiological functions of the liver. Second is selectivity. The coating blood vessels of the liver could cause side effects, and coating the blood vessels of the target tissue may prevent the transfer of gene therapy drugs. So we considered using a block copolymer consisting of long polyethylene glycol chain, the molecular weight of 8 kD, and short lysine or ornithine chains to satisfy these conditions. The oligolysine or oligoornithine chains are cationic and expected to bind to the scavenger receptors and proteoglycans of the sinusoidal wall. Furthermore, we tested actually the both one-arm and two-arm PEG chains. And I will explain why we compare these two polyethylene glycol with different configuration. So I will show you selectivity. That is an extrahepatic vessel observation. So we observed whether they would adsorb to the vessel walls of connective tissue outside the liver using in vivo confocal microscopy. Actually, we are now observing earlobe blood capillaries. Now, as shown here, neither the two-arm nor the one-arm polymers adsorb to these connective tissue vessel walls. It shows selectivity, no coating on connective tissue capillaries. However, oligolysine without PEG significantly adsorb to the vessel walls, so highlighting the effectiveness of PEG chains to elicit selectivity. Now, next, we look at adsorption to the sinusoidal walls of the liver. Now, for the one-arm polymer, sinusoidal walls are uniformly beautifully coated, as shown here. However, adsorption persists even after 9 hours, raising safety concerns. However, for the two-arm polymer, although it initially adsorbs very well to the sinusoidal walls, then it disappears slowly after about 6 hours, and likely gathering around the liver's bile area, suggesting slow excretion through the bile. So to confirm whether it's indeed excreted from into bile, we performed co-localization experiments with a bile tracer, as shown in this figure. As you can see, there is significant co-localization occurs. Therefore, as illustrated in the scheme below, this two-arm polymer adsorbs to the sinusoidal wall for a certain period, stealth coats the wall, and is then slowly excreted through bile. And looking at the blood circulation profile here, the PEG polymer without oligolysine chains remains in the bloodstream for a very long time, because this is a PEG with 8,000 molecular weight. Also, the one-arm polymer remains for quite a long duration, but the two-arm polymer is cleared from the bloodstream relatively smoothly. The difference between the two-arm and one-arm polymer is that one-arm polymer binds more strongly to the sinusoidal wall. Due to time constraints, I can show data demonstrating this later maybe in Q&A session if you are interested. So let's see how plasmid DNA loaded polyplex micelles behave with such a coating. In this figure, we use luciferase encoding plasmid DNA as a reporter gene. As you can see, the coating significantly increases blood retention and greatly reduce adsorption to the sinusoidal wall here. On the other hand, luciferase gene expression in the target organ, in this case tumor, increases significantly. And this transient stealth coating by the two-arm polymer has been observed not only in polymer micelles, but also in LNP and even viral vectors. Here, this slide shows the results for adeno-associated virus 8, AAV8 targeting heart and skeletal muscle. And this AAV is proposed for treating muscular dystrophy. However, hepatotoxicity has been a barrier to its clinical applications. So we administered our coating agent systemically here and followed by a systemic administration of AAV8 5 minutes later and observed luciferase expression in various organs. Now, expression significantly increased in the target organs, heart and muscle, while non-specific expression in the liver was greatly reduced. So our strategy may overcome hepatotoxicity issue of AAV-based therapy. I believe many of you here are interested in systemic administration of mRNA, messenger RNA using LNP. So this result here is from the systemic administration of LNP loading luciferase encoding messenger RNA and formulated according BioNTech's formulation. Now, mice transplanted with melanoma are used here. And you see high expression here in the liver as well as in the liver. That is a problem. So here is a, after coating, and you can see there's almost no expression in the liver. So there's a significant reduction in the in non-specific expression in the liver, and the expression in the tumor is identical. So here is a quantitative comparison of expression in the liver and tumor. The coating improves selectivity by more than 100-fold. The two-armed PEG oligocation polymer introduced in this presentation has already established GMP formulation and is currently already used in phase 1 clinical trials for systemic administration of antisense oligo and siRNA. So its use in clinical trials for messenger RNA therapeutics is expected in the near future. So if anyone here is interested in using this polymer, please get in touch with me. So finally, I would like to express my sincere thanks to all my workers and funding agencies, and happy to answer any comments and any questions. Thank you very much.